Thank you so much for inviting me. It's been a really exciting day for me, especially since my work is really far away from what has been presented today. And I invite you to get, in the next hour, next half an hour, you're going to be carried to even farther than electrophysiology to stem cell biology, developmental biology, and cellular phenotypes in patient-derived neurons. And so my lab is, the interest of my lab is really in developing cellular models for psychiatric diseases, and in particular for neurodevelopmental disorders, and autism and schizophrenia are our main focus. But what I will try to show you today is, tell you two stories, and one is related to psychiatric phenotypes, and the other one is related to a tool that we've been developing in the lab. But before I really start going into that, I really want to emphasize, the problem that we really have in psychiatry, which is not really the case in oncology and in neuro-oncology, just to see how far, behind we are, which is, I often say that in psychiatry we're suffering from symptoms of OES, and OES, I coined the term, but it stands for oncology envy syndrome, and it's really you can see that how behind we are in psychiatry, where every single disease is behaviorally defined, where we have no cure whatsoever for any of our diseases. We only have temporary patches. And we have no intelligently designed drugs whatsoever. We only have about 5 classes of drugs that have been discovered by chance. And so, why is that? Why did this happen in psychiatry? Why is psychiatry so behind oncology and neurology? So I guess there are multiple explanations for this, but the one that I like to emphasize today is that, well, if in the last 150 years since Emil Kraepelin really started to describe psychiatric diseases, we learned so much about the behavior of these diseases, and then in the last 20 or so years, we've learned so much about the genetics. The truth is we know almost nothing about the cells and the circuits that are affected in these diseases, and especially about the cell. So if you were to pick, for instance, autism, which is my main field, and if you were to ask, top researchers in the field, what is the cell type and the phenotype for autism? You would get as many answers as researchers out there, starting from GABAergic cells of the cortex to cortical excitatory neurons to even the hindbrain and so on and so forth. Not to mention that the phenotype is even more nebulous, right? Are the cells just synaptically impaired, or, are they dying, or, et cetera, et cetera. So what we're really missing in psychiatry is making, identifying the cells and the circuit. And that's mostly because we do not have access to brain cells from these patients. So my lab is really working on, essentially building human cellular models, or, what we sometimes call the mini brains of these patients in a dish. And so, I think the best illustration of really how, what is the pipeline of research in my labs has actually been made by one of my first patients with autism that I saw when I was still in medical school, who, when I opened my lab earlier this year, sent me this drawing of how he thought research in my lab is really carried. And so he thought that, what we're doing is, and I think he had a very neurosurgical oriented perspective. Right, so he thought we're probably climbing on this ladder, and we're poking some holes and looking through telescopes inside the brain to see neurons and, surprisingly, glia. Right, so he even knew about glia. So he's relatively high functional. And so I called him back on Skype and I said, look, it's you're very close, but it's not really exactly how we're doing this. So a couple of days later, he sent me another drawing, which I think is pretty much, illustrating what we're doing. So he said, what I think you're doing is you're taking skin cells from some of these patients, and then you're doing this mumbo-jumbo of transforming those cells in cells that very much look like stem cells. And because stem cells are pluripotent cells, you can make any cell type that you want, and I know you're making neurons. And so I think this is, this is what we're doing, and you can look at this also through a more professional schematic, but this is the pipeline of research in my lab. So we start with skin cells from these patients, and we reprogram these skin cells into, most of you have heard, induced pluripotent stem cells, which are cells that by all means look and behave like stem cells, but they retain the genome of the subjects from which they were derived, and then we take the skin cells and we convert them into neurons through various protocols that we have developed in the lab, with the hope that we will identify cellular phenotypes in these cells and ultimately find drugs. And so, just one introductory slide on how we're doing this, although this is becoming relatively standardized nowadays. We usually start with fibroblasts that we collect from these patients. We usually fully phenotype these patients clinically. We fly them to Stanford. They spend a couple of days, and then we collect their skin cells, and we put in, before we used to use retroviruses, now we use non-integrating means of delivering genes that, within a month or so, transform those cells into this large colonies of cells. I'm not sure if it's visible here. They're grown on a layer of feeders that very much look like embryonic stem cells. So these are the induced pluripotent stem cells. And then we'll go through a very long process that takes several months of really making sure that they are actually pluripotent. So, just to mention, what we're doing is, for instance, we're making sure that they express proteins that are expressed by embryonic stem cells. We make sure that their genome is fine, that they have not lost pieces of their DNA. We make sure that, those viruses, those episomal vectors have been inactivated. They're no longer expressed, that, by transcriptional profile, they look like embryonic stem cells. And we used to do this, where we're no longer doing it, is the teratoma assay, where essentially you take some of the cells, you inject them under the skin of nude mice, you wait for about 10 weeks, and then you look and you hope that you will see derivatives of all germ layers, which is really the golden standard for proving that a cell is a stem cell. So this, usually if everything goes well here, 100% of the cells are going to be pluripotent. The only issue is that this costs about $10,000 for each line, so it's becoming obsolete. The entire process anyway costs about $4,000 at least at the moment. And so what do we do with the stem cells? Where, we tell them that, they can be anything that they want to be when they grow up. And what we want to make them is, is transform them into neurons. So we and, the entire field has been really sort of piggybacking on what has been discovered in developmental neurobiology for the last 50 years or so. And so we're trying to sort of get clues from what people have learned from in vivo development and sort of try to mimic that in vitro. And most of the protocols are really laborious and are extremely long, and they require a lot of expertise, and takes at least 6 months of training for any of the postdocs that join my lab to really do this very reliably. And so, the process is so this is done in monolayer. All the classic protocols are done in monolayer. So you start with the iPS cells, and then, use MAT inhibition, you plate them down, and they form this rosette-like structure, which are essentially neural tubes in sections, like these beautiful flowers, that we collect mechanically under the microscope, and then we expand, and then we plate them down, and then we put growth factors. And so it's a really long process, and you end up with about, after about 5 to 7 weeks, you end up with about 40 to 50% of the neurons firing action potential. So most of these neurons are actually immature. You don't have astrocytes usually around them. So there are many limitations to this. And so this is how they look in culture, but there are multiple limitations to this system. And so, it's quite laborious. The neurons are not very mature. There's no cytoarchitecture to this, right? They're in monolayer, so, we're trying mostly to focus on making a cortex. And it's always, the cortex is a highly organized structure. There's no cytoarchitecture whatsoever in monolayer, even if you get some of the cortical neurons of various layers. There's usually no astrogenesis, so you're not getting astrocytes, which are usually formed at the end of neurogenesis. Because there are no astrocytes, there are no real synapses, and they're definitely not functional. And, there's barely any network activity. So how can you get from this to this? And I think we've been working really hard to develop a novel model, which is very different from what you've probably have seen around, but it's very similar to to what you have seen the organoid approaches, but because this is extremely defined, but it's similar to what people have been doing in terms of growing tumors in culture. So. anyway, we called, we're essentially building a fully, functional multi-layer cortex in 3D. We call it a spherical cortex. And it's sort of like talking about math, because there were so many discussions of the math, about math today. Sort of like, using the paradigm that mathematicians like. If you want to study a cow, imagine the cow, as a sphere. So it's sort of like you want to study the cortex, imagine the cortex as a sphere, sort of like simplifying. So the process it's quite simple. Actually, I'm not going to go into details. I'm just going to show you sort of like, and I can tell you more afterwards. We still start with the same iPS cells, but we have a cocktails of growth factors, and we have some special plays that we're using to get something like this. So after about 60 days, we're getting this perfectly round structures that are not attached, they're floating, they're not embedded into any extracellular matrix whatsoever. They grow up to about 4 millimeters in size, and they reach 5 after about 100 and something days. We can grow actually hundreds of this in a dish, so it's easy nowadays to make about 500 of this in one differentiation. As you can see, they're all around. They look very similar, and I guess this is as close as one can get to the title of the session that we had today, which was like Star Wars and New Technology. So I sort of like realized that actually this is the closest that I could get is actually to Star Trek and maybe New Technology, because I don't know how many of you remember, but there was actually an episode in Star Trek that was called The Providers, in which Captain Kirk at one point fights this super intelligent species, only to realize towards the end, walking into a cave and realizing that there were essentially 3 brains behind this entire procedure. And he has this really funny conversation that I just put here. He says, oh, primary mental evolution, incredible. At which point, actually the pulsing green brain says, that is not true, Captain. Once we had humanoid form, but we evolved beyond it. At which point the yellow one says, well, through eons of devoting ourselves exclusively to intellectual pursuits, we became the physically simple, mentally superior creature you see before you. At which point he says, a species that enslaves other beings is hardly superior, mentally or otherwise. Anyways, I guess it's as close as we can get to Star Trek as the title. But anyway, going back, so what do we really have inside this mini brains, or we call them cortical spheroids? Well, we did a lot of work trying to map them transcriptionally to the human developing brain, and luckily now we have this large collections of close to 2,000 samples across the human development, and we can really see where are we mapping, where are we, how far do we get, and it's actually quite surprising. While monolayer protocols usually reach, are in terms of similarity, they're mostly, they reach up to like 8 weeks of post-conception weeks. What we get after about 76 days is close to 19 to 20 weeks post-conception. And now we know, and the data is not here, but after about 130 days, when actually corticogenesis is finalized in the sphere, so it's really, you reach all the layers, they generate all the layers. At that point, we're actually most similar to a 35 post-conceptional week fetal cortex. You can also answer the question of where exactly you are in the brain at different time points, and we've been using this computational tools. I'm not going to go into it. So, if this is the developing cortex, for those of you who don't remember, the cortex really develops in an inside-outside fashion. So you have a large ventricular zones, especially in humans, and then neurons are being generated and positioned on top. In an inside-outside fashion, meaning layer 6 is made first after layer 1, which is an exception, and then layer 5 and 4 and so on and so forth. Right? So deep layers are called usually 5 and 6, and upper layers are 2, 3, and 4, and of course layer 1, which stands on the surface. So we're somewhere around here after about 76 days, and close to here after about 130 days. Now, if you actually look inside this spheroids, it's quite fascinating. So we really treat them like brains because they're big. So we fix them overnight. We section them exactly as you would section a brain. And so if you actually look inside these spheres, what you see are actually this large proliferative zones. So this is very early on with a ventricle inside, presumably. The progenitors are organized around it. They have perpendicular processes on top of this. There are neurons around that area. There's even a subventricular zone with TBR2 positive cells. And more importantly, for those of you who remember, most of the cells actually are very similar to glia. That's why they're called radial glia. They actually express GFAP early on. And they're the ones on top of which neurons actually migrate to the appropriate layer. So we actually see in these ventricular zones the perpendicular processes, they're GFAP positive, but they're still progenitors. Actually, we can isolate them, and you can see that they have two very clear processes. Most of the divisions actually happen in a very peculiar way. All of them happen actually close to the ventricular zone. So actually, the cells should, as they are in the brain, go down. The soma should go down when they're dividing, go undergo mitosis and then move up. And actually, just a couple of weeks, we managed actually to see that live and see this very specific type of division, which is called interkinetic nuclear migration. And essentially what, the cells are labeled here with the GFAP promoter with the virus. You can see this is actually the lumen. This is live imaging. This is one of the radial glia, and you will see what it does. This is a large ventricular zones, and you'll just see what the cell does. So the soma is really moving up and dividing, and then the cells are moving. This is very peculiar way of division, which you don't usually see. Now, if you look later, then you'll actually see that we do have a layer 1 type of, we have layer 1 neurons, which are usually positioned on the surface. Then if you look deeper, after about 130 days, you actually start to see lamination inside this brain. So actually you can see the ventricular zones here, which is like becoming, smaller and smaller. And then there are deep layers on the outside and superficial layers on the even farther. And this is done with multiple markers, has been highly quantified across time at different developmental time points. It makes sense from a developmental point of view. We also did a lot of work in single cell profiling of the cells, actually to show that we do have neurons of all the layers, and this is a panel of genes that have been recently validated by the Allen Brain Institute as being very specific for various layers. We can even, we've been even managed to sort of like, specify their, identify their regional identity, and most of them are actually prefrontal cortex and frontal cortex, and there are a few that are somatosensory and very few that are actually visual in terms of their gene profile. What is really actually cool about the system is that it does not just generate cortical neurons, but it actually moves from neurogenesis to gliogenesis towards the end. And so in between these neurons, you start seeing this GFAP positive cells, which increase with time up to about 20%. We've been doing a lot of reconstructions with the ray tomography inside this mini brains, and you can see a bunch of processes of these neurons in red here, and then in cyan, the GFAP positive cells. You can actually isolate them, and they really look like astrocytes that are non-reactive. They're star-like cells. And if you put serum, they become reactive. And we think that because there are these astrocytes, actually the spheres are quite active. So if you just open them essentially and you load them with a calcium indicator, without any depolarization whatsoever, you can see something like this. So there is network activity inside the spheres. This can be quantified. We've been doing patching in the cells, and what I think it's most important is that we actually see synapses that are being formed intrinsically as astrocytes are being formed. And you can even look here that we reconstructed one single synapse with multiple markers to show the perfect positioning of pre and post synaptic markers. But I think what is the most important part is the synaptic activity, really showing that the synapses are actually functional. This is work done in collaboration with Ben's lab and actually John's lab at Stanford. And so we're doing a lot of electrophysiology showing that there are spontaneous synaptic events that are glutamatergic that can be blocked. More recently, postdoc in John's lab managed to actually treat these mini brains as actual actual mice brains, and actually slice them and do slice physiology in a relatively intact structure. So you can actually record and stimulate in the cells and actually see a response to the stimulus, the electrical stimulus. In the cells, which we think it's quite It's quite an improvement versus anything else that has been done before. And then, you know, there are multiple how much more time do I have? Like I have 5? Yeah, maybe. You know, there are multiple applications that we've been working on this. You know, one of them is actually, you know, because we have the astrocytes and because we have the neurons, we really thought, for instance, to see whether we could develop a gliosis a model. And, you know, I'm originally from Transylvania, so, you know, people usually say that I have this tendency of, like, doing impaling experiments. But, you know, essentially what we did here, we took an electrode, a platinum electrode, and we simply sticked it through one of these mini brains. We let it there for about 2 weeks. And then, if you look inside about 2 weeks later, you can see where the electrode was. But what is really beautiful is the fact that most of the astrocytes have actually moved around around where the electrode was. Well, this is surprising for a few, you know, for at least 2 or 3 reasons. First, it is a human model. Second, it is an in vitro model, and you know, one of the biggest issues that we have, and I've been working closely with a professor in chemistry at Stanford, has been developing all these gorgeous materials that could be, you know, that she was hoping would not trigger gliosis, but there's no way she can systematically test them. In mice, it takes 6 weeks. It's incredibly expensive and very often non-translatable. So this in vitro human model, which is relatively inexpensive, can be quite powerful. And third, which, you know, the third issue that really shows that this is important is, well, there are no blood vessels here, right? I mean, there are no blood vessels, there are no microglia, they're just astrocytes, neurons, and progenitors. And you know, the classic model says that, well, you put the electrode in and you somehow harm a vessel and, you know, blood gets in and that triggers this entire process. Well, here actually we're showing that all you need is actually astrocytes and neurons and the electrode to actually trigger this. So, you know, there is power to this reductionistic model. Other things that we've been doing is trying to integrate this with other cell types. So for instance, we've been successful in the last few months to put any cell type on the surface of this sphere. So for instance, we can take oligodendrocytes that we collected from the fetal cortex, from the human fetal cortex, and just coat the surface and see how they're moving. And actually, it's really interesting because as the spheres produce a very thick extracellular matrix, and most of the spheres, actually, most of the dyes that we've been using do not get in, but oligodendrocytes get in within 12 hours, like quite well. We have no idea why that is happening, but they definitely get in. We've been also trying to coat them with endothelial cells that are also derived from fetal cortex to sort of like build on the surface a blood-brain barrier, and then that's still work in progress, but in principle it should be doable. And you know, anyway, so I guess I won't have time, right, to show. You know, maybe I'll just show you one example where we've been, you know, what we've been using this, not necessarily the system, but in general the approach for modeling disease, and that's in a form of autism, which is monogenic. It's very clearly defined, but it's very penetrant. So 80% of these patients that have this mutation actually have autism, and so it's called Timothy syndrome. It's extremely rare. It's actually a mutation in the calcium channel, which causes long QT syndrome in these patients, cutaneous endectly, and as I was mentioning, autism spectrum disorders in 80% of these patients. And this is caused by a mutation in the L-calcium channels, the ones that are hit by nimodipine and nifedipine. And so what we actually did, you know, and it's impossible to study this disease because, you know, you need the excitable cells. These are voltage-gated calcium channels. There's no way you can study this disease unless you have cells that that you can actually depolarize. So, you know, we made neurons from these patients that we characterized, and we observed, like, very early on a very severe phenotype in the cells in terms of the calcium. So when you depolarize the cells, there is more calcium staying in the cells after the depolarization than in the controls, and this is completely blocked by nimodipine, showing that it's a gain-of-function mutation mediated through this L-calcium channel. This was sort of like expected in a way, but we did a lot of gene expression, and one of the things that we have actually found was that the patient patient neurons were producing about 7-fold more neurons that were expressing tyrosine hydroxylase. And we spent a lot of time actually showing that this was ectopic expression, and that actually the entire pathway was functional to the extent that the patients were actually producing more noradrenaline and dopamine in a dish, although they were not supposed to, because these are actually cortical neurons, so they should not have tyrosine hydroxylase. And we identified a compound called roscovinin that was capable of rescuing these phenotypes within 48 hours. And we did a lot, we identified other phenotypes too that have to do with the corpus callosum. But another exciting phenotype, and this is, I'll finish here. Another interesting phenotype in the cells was actually a dendrite activity-dependent depolarization. So usually when you depolarize neurons in a dish, they tend, at least in the beginning, to extend their dendrites just because there is activity around. So you can see that this is what happens usually in control neurons. But if you do this in patient neurons, there is actually a retraction of their dendrites. So why is this important? Well, because this is a phenotype that you can trigger in a dish, and it's very predictable. So there's no difference at rest between patients and controls, but you depolarize them, and within 90 minutes you have this consistent difference between. So we thought it would be the ideal phenotype to actually devise a drug screening in a dish. So we built this platform for plating the cells. In addition, actually, had like 2 very good high school students who actually quantified this semi-automatically for for a couple of summers, hundreds of neurons that they traced. In addition, sort of what we found is that we found, for instance, that's how we found actually the roscovinin compound that I was showing you before, capable of rescuing. The problem is that roscovinin compound is quite toxic. It's actually in phase 3 clinical trials for small cell carcinoma because it's also a CDK inhibitor. But when we published the first paper, a company in France contacted us and said, well, all we do is actually make roscovinin derivatives. So if you want some. So we got a bunch from them. And so the second screen was actually not, to identify another compound, but can we find a compound that has the same effect at a much smaller concentration, which has a chemical structure that would not allow it to, for instance, have the side effects, because some of them are inhibitors of CDKs. And so doing the screenings, we actually identified 2 compounds that are actually, have the same effect in the nanomolar concentration, and which seem to be less toxic. So with this I had another example, but I hope I sort of convince you that that was another example, that I sort of convince you that you can use these models first for for identifying phenotypes, but also for devising in vitro drug screenings, or at least repurposing of certain drugs. And so with this, I'll end with this slide, which is, trying to apply this technology to the Gardner cycle of innovation, which, as you, all know, the prediction is that whenever there is a new technology, there is always a peak of inflated expectation, and this happened in 2007 with this technology. There were close to 200 labs that started doing it until they realized that it was incredibly expensive and extremely difficult to do. So I can tell by 2010, less than 10% of those labs were still doing this type of work. And I think with improvements in reprogramming, and now slowly in differentiation of the cells, and sort of repurposing whatever has been identified in chemical systems biology and in screenings, I think we're slowly moving on the slope of enlightenment and the plateau of productivity, which I hope is going to happen in the next year to come. And with this, I would like to acknowledge many of the people in my lab, as well as my close collaborators at Stanford, at UCLA, in Korea, and at the NIMH. Some of them providing patients or just doing experiments together. My sources of funding, and my newest member of the lab, my son, who just started pipetting. So I'm very excited that he's at 8 months. He's ready to start his first molecular biology experiments. And thank you very much.
Good reasons for this, but why not mix, oligodendroglial cells that you're producing with at the same time, rather than waiting this long sort of period for the neurons themselves? Is there some sort of reason they don't mix well, or they don't sort of deal with?
Well, we're not making them, so we were unable to make actually oligodendrocytes. So you can actually add them at any time that you want. We were just adding them towards the end. The astrocytes are generated intrinsically, so they make neurons, and then they suddenly just move towards making astrocytes. they should theoretically move into becoming, into generating oligodendrocytes. It seems like we probably are either missing something or they're not doing. So we're adding them towards the end. Actually, in a way, my biggest fear was that they will make oligodendrocytes, because oligodendrocytes are going to myelinate. And so that lipid, it's going to, is just going to make it impossible to do a lot of experiments. And so, the approach has been to sort of like put them on the surface, which also, another advantage is that we hope, and that's what we're looking at, that is going to create an in vitro model for myelination on the surface, a human model for myelination on the surface.
Right. I can tell you when we were making spheroids of tumor cells, we tried to put the endothelial cells on the surface many times, but unfortunately for some reason they weren't able to completely get into the center and deal with the spheroids as far as tumor cells are concerned. So there may be some differences that you have to look at.
Yeah, so the first thing that we discovered actually with endothelial cells was that they hate the neuronal media. So they, we try for a few weeks, and that was actually my own experiment in the lab. I usually try to have my own project. And I was doing this with the tech in my lab, and I kept saying, okay, well, you're doing something wrong. It just seemed like there were no endothelial cells on the surface whatsoever, until she did secretly the control experiment, where she just put the neuronal media on top of the endothelial cells just to see they're all dead within 24 hours. So apparently they need serum, which we don't use here, because the moment you put serum, the astrocytes actually, we discovered, move on the surface. And they become reactive, and of course neurons are dying. And so now we discovered actually that if we do that only for 48 hours, it's still not that bad. So astrocytes are actually moving on the surface, and we're hoping that actually it's one of the first steps in creating a blood-brain barrier on the surface, because they're actually going on the surface. And so with that, we don't know yet what it's precisely the effect on the neurons, but at least for 48 hours, the spheres still seem to be fine. And endothelial cells attach, and at that point you can use whatever media you want, presumably, because it's not getting in anymore. But we're still sort of like working that out. That's complicated.
Well, this is very novel, and I congratulate you in creating these brains on a dish. I have a couple questions. One is that if you just leave them on the dish, do they grow forever, or is there a limit as to what they grow? Is there contact inhibition? Also, another thing is that in our brain tumor universe, there's a point mutation called isocitrate dehydrogenase mutation that is important, and we think that it occurs very early in gliomagenesis. Can you create point mutation?
Yeah. So the first question is about the size. So we did notice that they stop at around 5 millimeters, which is sort of like what I guess people have been observing in the tumor field too. That's what I was told, that there's always a limit because we don't have vasculature. My biggest fear was always that inside there's going to be not, I guess the neuropathological terms would not be necrosis, because there's not really necrosis, but just cell death. So it's surprising that there's actually no cell death in the middle. A lot of people told me, oh, there's going to be hypoxia. So we stained actually, because somebody in my lab is trying to develop a premature brain hypoxic injury model. And actually, there's no hypoxia whatsoever. There's no cell death in them. So it seems like, when they reach 5 millimeters, they sort of stop, and they don't die. What we can do to make them bigger is actually, they're still epithelial cells, so they love to attach to anything. So if you put them close to each other, they fuse easily. So you can actually generate very large mini brains that are composite of, multiples. Even these chains. So actually we have to be very careful when we're growing them, because otherwise they attach to each other and they create these chains in a dish, which we're actually now leveraging in a different, so this is just an excitatory cortex, and you know that inhibitor neurons are actually made in a different part of the brain. So we have no inhibitor neurons whatsoever in this, and so what we've been doing is actually patterning them separately into becoming a striatum-like structure, or a medial geniculate eminence-like structure, and then we can, fluorescently label them. We can put them close to each other, and they fuse. So they merge initially, and then within, a week or two, they're completely fused into a larger sphere. So that seems to help. I have no idea what the consequences are in terms of like the survival inside, but the ethical implications too, I guess. Yes. My chair is actually an ethicist, and so far she hasn't raised any. I'm just like waiting for her to point whenever it's like we're going too far. But Yeah, so essentially you can engineer one piece of the brain at a time and sort of like merge them. And 5 millimeters seems to be the size that they're like comfortable with. And the second question, yes, we've been using CRISPR actually, so to induce point mutation. Of course, I think it would be very difficult to induce them at a very specific time point, you know, as they're progressing, because CRISPR technologies are still like, you know, quite difficult to implement in pluripotent stem cells. But you can definitely take the same, for instance, control iPS line and, you know, induce that mutation and sort of differentiate them in parallel. That's absolutely possible. Yeah. Okay.
Great. Thank you.
Okay. Thank you.